All right, I'm live, apparently. And my health is good. Okay, welcome everybody. Good to see you all. See some of you in person, some of you online. Uh, I guess you're all online. None of you are in person. Okay, I can see some people. uh, anyways, oh, and then I guess just 1 second. You want to see what's going on, right? So I don't want to make you do that. Okay, just 1 second. There's a little bit of workout things I need to do. Where is it? Oh, there it is. Sorry, just 1 second. You'll be getting. All right, I'm gonna restart this call in 1 second. So. Okay. Sorry, everybody. We just oh, I just probably killed my stream, didn't I? Force quit. Okay. I'm not sure if my stream went down or not. So just my browser went down. Might be chopping a whole bunch off the end of the stream here. Okay. Okay. I do have my, okay, my stream is live. I'm sorry about that. Just 1 second. I'm just getting some other things sorted. My YouTube, I had to close it, and now I'm just managing things. No, I want to go there. Okay, so today I posted some videos that I'm just going to get started again. Okay, here we go. There's a call. So many things going on. All right, I'm gonna give up on the call. Oh, that one was working. Okay. Oh, wait, close. Click to join. There we go. I'm the only one here. Oh, that's weird. You are in this call. Nothing more exciting than watching someone troubleshoot for the first 10 minutes of class. Okay. All right. I think I've got everything going. Now I'm gonna just get, oh, I see your comment now. Let's see, I just want to see if I can go all the way on this this thing and screen share and all this sort of stuff. So I'm gonna use an application window, screen one, application window, and I want to pick this application. I'd like to give it an application, share, your screen sharing. How does that look? Is that screen sharing the right thing? Well, okay. I will make that full screen, there. And now, all right. How many different things can I have going on at the same time? Oops, I don't need this. Okay, that's, okay, so it's sharing the whole window, which is not exactly what I wanted, but it's, that is not show, well, let's see what the preview looks like. Okay, well, kind of halfway, but not quite there. No, I want this to be up front, and then that'll cover you. Okay. All right, so I cannot see the chat right now, but I can see everything else. So hopefully you can see what you need to see. Oh, okay. Just gonna do this. And then, all right. Okay, close enough. Okay, get rid of the preview. If you want to join our call, there's a call on the Hangout. I just found out today that that Google Classroom has now incorporated Google Meet. So we will probably switch to Google Meet in the future, and then I believe I can do a recording in Google Meet of the classroom, and then I'll just upload the recording after class instead of streaming it, I think. I think that's what I'll do. If you have any strong feelings about that one way or the other, then don't worry. Then feel free to share that with me in one format or another. It's just going to bring a couple of things over here so I can see them and see how that goes. Okay. And all right, so now everything's on my smaller screen, but that's okay. And there, the call froze, but the audio is working. How's everything going right now? Is everything look, everything's working okay? Excellent. Okay, that's a good test too. I'll probably cut this first 10 minutes. I'm gonna just make a note that 12:10 is when I want to really get the video started. Okay, so Mendelian genetics. I just want to do one bit of recorded coverage of the dihybrid crosses before we move to the next subject. So this is going to be our recorded version of the dihybrid cross. And I've sent you a little PDF of some questions I've done in the past, ones that I only had in PDF form. So this is a scan, which is my least favorite type of file, but that's okay. So in rabbits, black color is due to a dominant gene and brown color due to a recessive gene. So we are using a capital B here to represent the dominant gene and a lowercase b to represent the recessive gene. Short hair is the result of a dominant gene, which we will call S for short, and long hair is a recessive allele. The recessive allele has to retain the same lettering, so it is a lowercase s. What would be the genotypes of the F1 generation if you crossed a homozygous black long-haired rabbit and a homozygous short-haired rabbit? So in this case, we need to first identify our parents. So parent number 1 is going to be homozygous black hair. So that means that they must have 2 dominant alleles for their dominant genes, and they are long-haired. Now notice that in long-haired they did not give us any allele notation. They didn't tell us if it was homozygous or heterozygous, and that's because earlier on we said that the long hair is the recessive gene. So by being the recessive gene, if something is long-haired, it must have the homozygous recessive genotype. So that's parent number 1. Parent number 2 is going to be homozygous for brown hair. So that's excessive because we know that brown hair must be also the recessive genotype. So that's going to be 2 little brown hairs. And they are a short-haired. And I think this is where, every year I use the same file and realize that there's an error here, that it should actually say, it should be a little bit more specific that it's homozygous for brown and short hair. So short hair being the dominant genotype, we're going to make that assumption that the homozygous refers to that as well. Oh, hello everybody on the call. It's great to see you. This is also streaming to YouTube, but the YouTube does not see your faces. So only if you talk, you might be heard. We haven't quite figured that out yet. So then the next thing to think about is what sort of allele combinations are possible for each of these parents. And then we're going to take those allele combinations and place those on our giant Punnett square. So some allele combinations we're going to get for parent number 1. Well, you have to choose one allele from the one B allele and one S allele. There is only one type of B allele, and there's only one type of S allele. So in fact, all of them are going to be BS. So these are the only possible combinations of alleles that you can get from this first parent. The second parent is also going to create gametes. Those gametes are going to have one copy of every allele. So they will have one copy of the B and one copy of the S. But in this case, the parent only carries lowercase b's and uppercase s's, or only carries recessive b's and dominant s's. So all four of their combinations are going to be of this setup. So we need to set up a Punnett square. In this case, some of you that are quick on the draw will realize that you actually don't need much of a Punnett square to do this, because you have 4 situations that are all going to be identical, creating 16 identical crosses. But let's do all the work just for fun, in quotation marks. And I also made one extra column, which is what happens when you talk and try to teach at the same time. Use a darker color. And so the other parent is going to be down here. And so you should see that that any cross, any square that I try to fill, I will end up doing the exact same cross no matter what, because each parent only carries one type of each allele. Each parent is homozygous for each, so there's really not much, there's no probability here. There's only one possible cross. And this is really what's happening in the case of, say, pure breeding parents, is that they might be able to just do this sort of crossing because they only carry one type of allele. I'm keeping my eyes open on a couple of different things here. If I haven't quite figured out how to open the chat window for the Hangout. So that's the only thing I'm not watching. I am watching the chat in the Google Stream. If you want to just talk in the Hangout, then I will hear you. I guess I can open the chat on yet another device here, and see how that goes. There we go. Okay, I do have my chat open as well. So all of these are going to add up together, and we're going to get something that looks like this. And we can carry on. You could, just with a quick copy and paste, you could fill this in. I'm also comfortable with you doing something that looks like this to just show that all of these are going to be identical. So what is the genotype of the F1 generation? The F1 generation is going to be BBSS. They're going to be heterozygous for both. And so heterozygous, or carriers for both traits. And that means that they are going to have the dominant phenotype for both traits. So they will be, what is it, black-haired? Yeah. Black and short-haired. If one of the F1 rabbits was mated with a brown long-haired rabbit, predict the genotypes and phenotype ratio. So we now have our F1 generation, which looks like this. So that's going to be parent 1. And our parent 2 is going to be a brown long-haired rabbit. So those are both recessive traits. So that means that they must be in this type. So what sort of allele combinations can we get? So this is the one where we have to be a little bit more careful. We're going to use the outside, the first pair first. So a capital B, a dominant B, and a dominant S. We're going to use the outside, dominant, and a recessive. We're going to use the inside, recessive, and a dominant. And we're going to use the last pair, which is the recessive and recessive form. If we do the same process with parent 2, of course, because they're homozygotes, all of their allele combinations are the same for all their gametes. So these are our gametes. And so let's get ourselves a little square going. It is fair game that if you have only one type of gamete coming from one parent, then you are free to simply do one column or one row. So we could simply just write BS like this and not do any more rows because all of the rows are identical. All identical. So any combination will be exactly the same. But that's more of like an advanced strategy. All right, so then we're going to carry through with our copying. I'm going to do all 4 just because that's what we've been doing up until now. And I don't want to make any new exciting changes here. Okay, so there's BS. So I'm going to take my allele from the top and an allele from the left, allele from the top, allele from the left, allele from the top, allele from the left, allele from the top, allele from the left, top, left, top, left, top, left, top, left. And then because all of these are the same, we are actually going to see that these are all identical. All the way down. So in terms of sort of probability-wise, we are looking at a setup where the probabilities are pretty much set to a 1 to 1 to 1 ratio here. The probabilities are reduced by 4 kind of thing. So we got a whole bunch of things going on. We're going to have 4 of these. We're going to have 4 of these. We're going to have 4 of these. And we're going to have 4 of these ones. And so in terms of genotypes, in terms of phenotype, we have dark and short. I'm just going to use my zoom here so I can write nicely. Dark and short, or black and short, that's what they called, right? We didn't say dark, we said black. Short, black, and long. I feel like that's the rabbit I would want. Or is it a guinea pig? It's rabbit. Yeah, rabbits are a little bit bigger than guinea pigs. I think they're a little nicer. We got brown and short. And we have brown and long. And in this way, we can think they were 4 to 4 to 4 to 4, but if there is a common multiple or a common divisor in your ratio, then you can reduce it down to the smallest common value. And so for 4, 4, 4, it's the same as writing 1 to 1 to 1 to 1. And so those would be the 4 genotypes and 4 phenotypes and their ratio. Now, I do like to follow the sort of provincial guidelines on this sort of ratio notation. In the provincial applied math exam, if you represent your final answer in any form that is correct, that includes ratios or percents or fractions, those are all equally accepted as correct answers. I agree with that. If you write 4 to 4 to 4 to 4, that's to me an equivalent answer as writing 1 to 1 to 1 to 1. In the future, you might be required to provide, provide simplification, but in my course at least, as it's only a very small portion of our course, I would just let you get away with whatever. As long as the ratio is correct, doesn't matter what format you used. I think that's just being, too picky. This day and age, we can't. This day and age, as in the current times, I don't think we should be as picky at least. All right. In watermelons, the genes for the green color and the short shape are dominant over the alleles for the striped color and the long shape. Suppose a plant heterozygous for both of these traits is crossed with a plant with long striped fruit. And in this case, this looks pretty boring to me. This looks like it's going to be exactly the same ratios as last time. You guys notice? And that's also kind of the thing that happens in these questions. So we have a heterozygote, right? So we're talking about a parent who is heterozygous for both. And they're going to be crossed with a long striped fruit, which means that they're getting crossed with the recessive genes of both. So in fact, this will be exactly the same question as the previous one we just finished. So I'm going to just skip ahead because I don't want to spend all day on these dihybrids. But you should be able to predict that we're going to get a 1 to 1 to 1 to 1 ratio. Okay, so let's see if this one looks a little more tricky. So we have, this one has always given problems with my students. They do not like the vestigial wings and hairy body as terminology. So in the body of the fly, which kind of, they look like this, there is a case where some flies are born with an extra pair of wings that are kind of, sometimes not even a full pair, but they'll get a little extra wing down here. And that's a vestigial wing. It doesn't actually help them fly at all. It's just a wing that some of them carry on them still. And yeah, and then hairy body is they actually have little hairs on their body. That looks like little legs, so maybe I'll make it a different color. And fruit flies have been used for a long, long time. If you watch that documentary, the Great Transformations documentary, you'll know that there's a big section about fruit flies in there and how they're very useful because they reproduce very quickly. You can make changes to their genome and things like that and watch them change rapidly. Okay, so the normal alleles are long wings and hairless body. I thought the vestigial wings were an extra pair of wings. They might actually be little tiny wings that don't do anything, like that. In our lab in university, they were little tiny wings that were a result of radiation. Oh, okay, great. So there we go. I never got to do that lab, so that's very exciting. Was that in a genetics course? Yeah, genetics 2000. Okay, interesting. Well, I made that course better now. I think there was no lab when I took it. Oh. Yeah, it was extremely dull. But it was yeah. Oh yeah, I bet. Yeah, there was no lab component. It was very very nothing. It was just sat down there and multiple choice exams, and yeah. Okay, well that's great. I'm glad that they improve that. They should. All right, so we have the normal alleles are the long wings and hairless body. So I guess we'll use a capital L for long wings. And then that means that we'll have a lowercase l for the vestigial. And then we'll make a long wings, and the hairless body, well, we'll just use big capital H for hairless, and we'll use a lowercase h for hairy. And so the actual cross here, we have a vestigial winged hairy male. Vestigial winged means that they must be the two little L's, and they are hairy, so they are lowercase h, is crossed with a female homozygous for the long wings and hairless body. And this again looks like a very similar cross to one that we did at the beginning of the unit, right? Well, I think that's the same cross we did in question 1, almost. It's almost the same cross. Well, let's carry through then. So for one parent, let's put the male here, we can only take lowercase l's and lowercase h's. For the other parent, we can only get capital L's and capital H's, so every single square is going to be the same. And then we carry through. And over there, you can carry them all the way through. All of these are all identical because they all have the same. There's another way of looking at this that's more mathy if you really like math. Let's see if I can do it off the top of my head here. So in this case, we can think about that let's look at this first parent. So right here, we have 0.5 or 50% are capital L, 0.5 are, or actually 100% are capital L, right? So let's think about this as we have 100% times L. You can think of it as 0 times little l. They only have 100% of the dominant allele. And for the H here, we have 100% of them are the dominant allele, 0% are the recessive allele. So what is the possibility of forming this allele combination? Well, this allele combination is 100% times 100%. So not very exciting. And then when we go to making combinations of combinations, we can multiply those as well. All right. So, but when we start considering this combination, so we end up with a combination like this, L, H, H, L. Now we can think about this, that they have, for L, there is now a 50% combination. For the lowercase l, you now have a 50% chance. And for the capital H, you have a 50% chance. And for the lowercase h, you also have a 50% chance. So when we consider this, then there's some more math things we can do later on when we think about, okay, so from here, what is how many of them have this form? Well, this would be 0.5 times 0.5, and that gives us 0.25. And as long as you write out every option, you will, always pressing on that space bar. Okay. You can work out exactly how much percent are going to be in the various forms. And then you could work on the next set if you wanted. If you cross two of these individuals with each other, then you could take, okay, well, what is the chance that this combines with this? Well, that'd be 0.25 times 0.25. That's smaller, 1/4 of 25. And then you could carry on and carry on. So there would be a mathematical way to do this. The more combinations you're making, the more difficult the numbers become. And yeah. Anyways, that's just excess. So the last piece here is that if the F1 generation in the last question were interbred, what would the phenotype ratios be? Well, then we would be talking about interbreeding this hybrid. And so I don't even have to finish the question. Hopefully at this point, you should just be able to go, the answer is 9331. There is going to be 9 individuals with the dominant for both, 3 individuals who are dominant for one trait, and 3 individuals who are dominant for the other trait, and then 1 individual who's recessive for both. So that is going to be the end product here, because this is the true dihybrid cross. Dihybrid cross. Okay, so let's see if we can prove that for ourselves. So as per the little bit of math I did here, I can see that 1/4 of my individuals will be of each type. So I have 1/4 like this, 1/4 like that, 1/4 are like this, 1/4 are like that. It doesn't actually matter what order you do them in. I think I might have just chosen a different order than I normally do, which is, didn't look like much of an H. Okay, so I usually do what? First, outside, last. So first, outside. Oh no, I did the right order. Inside and last. And so now I filled out the square, and then I can start to complete these circles. So this is the only one that's going to be dominant for both. This one will also be homozygous, but for 2 different traits or 2 different types. And this will be the homozygous for both recessives. And then we just start to fill in the heterozygotes here. Oops. Try to write them in the right order so it's easier to track. And carry on. And I believe we did a couple of these the other day, but I guess I should really finish this one. I get kind of bored about halfway through filling out these tables. I don't know about you. It's hard to keep my attention. Well, there is something a little bit peaceful about it. At least you know that there's only one answer. I do like that kind of aspect of these genetics questions, is that there's only ever one answer, so you have that sort of finality to the question, which can be comforting. Okay, so I've got these 3 here, which all have similar appearances. They have different genotypes, but they do have similar appearances. I've got these ones here, which have the similar appearance, different genotypes, similar appearance. And we have the rest, which have identical appearance, but different genotypes. You should be able to notice that things that are on opposite sides of the line here. So if you think about there being a line, that's sort of like a symmetry line. So you will have genotypes that appear here and here that are identical. This genotype here is the same as this genotype here. This genotype here is the same as this genotype here. They show up on opposite sides of that symmetry line. And a similar trick works over here. You can see that this is the same position on the opposite side of the symmetry. So that's always kind of, there's a whole type of math called space groups that plays on this idea that if you multiply things in certain ways and you apply, certain theories to certain sets of equations, then things will work out in these sort of patterned ways. So we can organize. We did establish our 9 green ones, which are our 9 dominant types. So that's our long. So these are 9 long, hairless. And then we got 3 that are going to be long and hairy. We got 3 that are going to be vestigial and hairless. And we got 1 that is recessive for both, so it's a vestigial and hairy. In terms of the genotypes, this is more tricky. It's a little bit harder to count them out, but you're going to get 1 of most, and then you should get 4 that have. You should get 2. You should get pairs for some of them. So let's see if I can find the pairs here. Oh yeah, look at this. I just realized that it's even better. These are all on the same line in the opposite direction, so they all have the same genotype as well. So notice that. Okay, so there's our 4. We get 4 that are the recessive, the heterozygotes. We get 2 which have this dominant with a heterozygote. We're going to get 2 that are heterozygote for the first and homozygous for the second. We're going to get 2 that are recessive for the first and heterozygous for the second. We're going to get 2 who are heterozygous for the first and recessive for the second. We are going to get 1 that is dominant for the first and recessive for the second. I don't know why I put a dot there. That's going to go. Okay, we are going to get 1 that is recessive for the first and dominant for the second, and we are going to get 1 that is recessive for both. Let's see if I did it right. 1, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Good. There should always be 16. So, and that adds up to 16. So for the patterning, we notice that if you're heterozygote, you always double the number of options. So if you're heterozygote for both, then you get 2 times 2, you get 4 options. And if you're only heterozygote for one of the traits, then you have twice as likely. Heterozygote for one of the traits, heterozygote for one of the traits, heterozygote for one of the traits. So that's where that mathematically that works out. And those are also the ones that show up on off angle. So that's sort of exciting. It's probably a degree of freedom notation there. That's probably right. Anyways, there's a deeper reasoning for that. Okay. In question 3, with long wings was crossed with vestigial. Oh, okay. Well, anyways, this question keeps going deeper. I would like to move away from this. Everybody's okay with that. Is everybody okay with these dihybrid crosses? I can update this sheet with some more answers later on, but feel free to play around with these questions if you need some practice, or we can have a meeting later if you feel like you want some help with that. Okay, so let's go back to our package. And we have non-Mendelian inheritance patterns to cover. So this is where things get more exciting. And for the majority of the remainder of this unit, it's all about exciting different new patterns and what those might mean. So the first one we're going to cover is called incomplete dominance. And so this is a pattern where the dominant allele, so I just want to get this going. So in incomplete dominance, phenotypes, as soon as I move the box, it doesn't want. Okay. All right, so in incomplete dominance, phenotypes that carry both the recessive, or both both allele types have genotypes that carry both allele types, so heterozygotes, have a third unique appearance. Typically an intermediate phenotype. So the most common one that we cover is usually the snapdragons. So snapdragons come in a white and a red variety. If you cross the white and red variety of snapdragon, you end up with a pink snapdragon. And that pink snapdragon has a unique phenotype. It's neither red nor is it white. It's something else. And in the background here, what's actually happening is that the two alleles seem to have some sort of relationship to each other, some sort of genetic activity that they kind of work together. Alleles are likely working together to form the trait. It's not exactly, but yeah, that's okay. So there's a couple of ways that we could imagine this might work. One way to think about it is that if you have cells that have these two chromosomes, there's this red chromosome and a white chromosome, or a chromosome that produces a red color and a chromosome that produces a white color. It might be that you have two chromosomes producing both colors, and then from afar, or from the beyond the cell level, these cells now appear to be red or white, red and white mixed together, and our eye sees that as pink. It might also be that red flowers use both chromosomes, both red producing chromosomes, to produce enough pigment to give it that red color. And then when you have a red and a white chromosome, the white chromosome is essentially not doing anything. And so the amount of red being produced is half as much. So I'm not sure exactly the mechanism that makes pink flowers. The second mechanism is probably the simpler one, where just having one of the two alleles just doesn't do anything. And so the result is you have half as much pigment being formed, and so the flowers appear to be pink. But either way, the flowers are influenced by both alleles being present. So that's really the trick about incomplete dominance, is that when the other allele is missing, when the other allele is different, you end up with a new appearance. So this looks like this type of deal. We are going to use this notation. If you want to shout in if this is a notation you've used before. Sometimes we see I've seen a couple of different variations where they might actually use another letter, or they might just say it's incomplete dominance and use the normal capital R and lowercase r. But a lot of the time, they'll go one step further and try to indicate this notation of the incomplete dominance by putting an apostrophe on the other allele, so that one allele has the capital letter by itself, the other allele has the same letter, but with an apostrophe to indicate that it's some sort of variant of that allele. So in this case, we have a red flower up here. This one is red, this one is white. And when they cross, we end up creating a pattern of two different alleles, a whole bunch of heterozygotes, just like normal. When you take two homozygotes and cross them, you end up with a set of heterozygotes. And so these four heterozygotes are all going to have the new allele appearance. They're going to have the new pink appearance. So this is going to be 100% pink. And this is actually makes a lot of questions easier. to be honest, because now you have a direct measurement for those heterozygotes. You don't have to guess about heterozygotes anymore because you have a direct visual component that tells you how many heterozygotes you have. So, for example, we can say that, we can say something like, if there are two, if we can see if we can count up these two pink flowers, then we know exactly that there were, we know exactly that their parents must have carried the recessive gene or the white gene, and we know exactly what the genotype ratio is here just by counting up the number of phenotypes. So the ratio of phenotypes and genotypes will be the same for an incomplete dominance inheritance. I cannot get myself to write inheritance correctly the first time around. Okay. And then the next type is going to be, and then we're going to contrast that with another one where the two alleles work together in a unique way, and that is called codominance. So in codominance, we have, codominance, in the heterozygous genotype in codominance creates a unique phenotype that has characteristics of both alleles, or has the traits of both alleles. In the cell, both alleles are functioning and creating and whatever. That's probably good. Okay, so this can be a tricky one to wrap your head around, especially if you're not used to breeding cows or anything like that. But there are quite a few examples of this out in the wild. So one example is the roan cow. So the roan cows is when you take a purebred white cow like this and a red cow and you cross them, and the result is a mixture of red and white in the cow. So they end up with, in this case, you can see the red splotching on their head and the white body. So they have skin cells here, or fur cells that are producing both the red and the white colors. And they are working together, and they're producing, and so both traits are present. Another example of this is in human blood type. So the human blood type example, human AB blood type. The cells of this heterozygote will be covered in both are they carbohydrates? They're carbohydrates. Both the A and B type molecules, so that the cells are identified by the immune system as both A and B. So this is a really unique feature of the A blood type. Probably not unique to humans or anything, but it is the AB blood type is a heterozygote codominant blood type. There are two alleles that are in charge of A and B, and when they work together, they form AB blood type, and that AB blood type has both the characteristics of A and B type blood. And so when they are identified by an immune system, the immune system will identify it as both of those types. Anybody have any questions about codominance? This is different from incomplete, because if we had incomplete AB blood, then when A and B came together, they would form C-type blood. And it'd be identified as a new, the immune system would identify it as C blood, and would have, it would be something in between, or there's no letters in between A and B, so I can't choose anything else. So it'd be C blood, and it would be something new and unique, and your immune system would have a new, unique way of responding to it. Instead, we have AB blood, and the immune system can use either A or B antigens to attack that blood type. It is both. We can imagine, there's probably other examples. I've heard of zebra being accused of being a codominant blood type, or a codominant effect. I'm still wrapping my head around it. I think it's like a codominance with multiple alleles. Otherwise, we would see all white and all black zebras out there, but that's possible as well, that they have both alleles, and that's what leads to their patterning. Which makes sense if it's multiple allele. If we think about well, human hair color is a little bit tricky. There are some examples of human hair color that are incomplete dominance, I suppose. Eye color is probably close to being an incomplete case, where having different combinations of alleles leads you to different eye colors, but there are multiple alleles in charge of your eyes. So that also makes it tricky. That's why we use plants a lot. It's a little bit simpler to work in. Okay, I think we're going to get too deep into the well if we go to the next one. The next one is the multiple allele system. So I think I'd like to do that as a separate lesson. Let's just do, do I want to do a practice question? Let's see if we can find one practice question that I can pull out here. And I had some pulled up earlier. Just one second. Boom. Maybe I will move this. Oops. So let's see. We haven't. Well, okay. We can just do one. So a red and a pink snapdragon are crossed. What is the ratio of genotypes and phenotypes? So if this is the question we're left with, we have a red and a pink snapdragon, so that means that we have one parent who is red, and they're being crossed with a pink parent, which is a heterozygote. And so we would be looking at doing a little simple cross with the one parent on top, the other parent to the right, and the result is going to be taken from the top and from the left, top and from the left, top and from the left. This is also really important that you're nice and professional with your approach. Make sure you're using capital letters in this case. Make sure you're including that little dash. That is important. And we see that we get 2 RRs to 2 heterozygotes, or 1 red to 1 pink ratio. If you wanted to create white flowers from this is a question I've actually very often asked students, is if you wanted to make white flowers, and this was your, this is what you were left with, what would be the best approach to making those white flowers? So you've got these 2 plants, and you want to make white flowers. What could you do in that case? I'll make white flower. Well, this is a little bit tricky because we are talking about plants and people might have their mind set on animals. And with plants, you do have an option of self-pollinating. So you could breed pink flower with itself, and the result would be a 1 red to 2 pink to 1 white. And then you could have a white flower, and then the white flower you could start to breed by itself, and you could get all white flowers from that point on. So that would be a tricky question. Okay. All right, I think that that covers that one. I guess we should do one. We didn't do an example here, so let's do a human. We're gonna do human blood type in the next class, but let's do an example here. A homozygous A type, an individual homozygous for A type blood has children with one who is AB blood type. What is their possible children's blood types? So very simple. I just realized I just created the exact same problem I did above. You can think about this as, we'll go through the actual notation tomorrow, but we are going to take our two A's, they're homozygous. We're going to take our A and B and cross them. Gives us two children who are A blood type and two children who are AB blood type. So 50% A type, 50% AB. All right, that's it for today. Let's take a break. Tomorrow, let's see if we can get a Google Meet going on. I'm not exactly sure. I will share the Meet code with you in Classroom, but apparently Classroom now creates their own Meet codes. So I will share one with you, and we'll see what that's like tomorrow, maybe. And we'll give that a shot. Thanks everybody for joining in. I know we just got these Hangouts working, but now we'll have another option. And the good thing about the Google Meet is I can leave the meeting open, and you can join and leave at any time, which means that I can tell you which office hours I have the Google meeting open, and you can just kind of pop in, ask me a question, and then pop out. So that'll be the next step, as opposed to the Hangout, which is good too, but yeah. Anyways, that's what we'll go next, and that'll be sort of an ongoing thing that you can also join in. I'm not sure if it's necessary, maybe. This is probably working fine. But anyways, we'll talk to you later, and have yourselves a great day.